Hello everyone, my name is Sofia and today I'm going to talk to you guys about biotin. I talked to you guys a couple lectures ago about riboflavin. So today I was asked to teach this lecture again. So I'm here with you guys one more time. So we're going to talk about biotin now, vitamin B7. We're going to talk about digestion and metabolism, mechanism of action, sources and requirements, deficiencies, and toxicities. So, again, we're still talking about B vitamins, the B complex as well as vitamin C are water-soluble vitamins. So the excess can get removed from our organisms by urine. So we discussed this a little bit about riboflavin, but usually B vitamins are considered like energy boosters. So here in these little shots, probably you've all seen in like the 7-Elevens or anywhere, the 5-hour energy shots, you see this claim, it's packed with B vitamins and amino acids. However, we're not sure if the energy boosts comes from the B vitamins or the caffeine or taurine or other stimulants that this energy shot has. However, of course, B vitamins are important in energy metabolism, but not to the extent of caffeine, of course. Again, this vodka here claims to have an energy boosting properties as well, and anti-hangover because of B vitamins. And I think this is completely exaggerated. Again, even though it has some effect on energy metabolism, it's not like the B vitamins is going to give you an anti-hangover vodka. So biotin, probably you've all heard about biotin because it's usually marketed as a supplement for hair and nails and skin. And a lot of people who have alopecia tend to take biotin. And it's a fairly popular vitamin. And like riboflavin, people usually don't hear about riboflavin. I'm talking about riboflavin because it's the one that I had to teach you a couple lectures ago. And I recorded them very, very close. So yesterday I recorded riboflavin, so I have all of that information in my mind. So I'm going to be referencing that lecture pretty often. Biotin is more common than riboflavin, definitely. So riboflavin was discovered in the 1930s, and actually this is quite an interesting fun fact. So there was and there is a disease that's called the egg white injury. So people who ate plenty of raw eggs experienced hair loss and dermatitis, some severe cases of hair loss and dermatitis. And then they realized that there was a substance that was isolated from liver that was shown to cure the symptoms of the egg white injury. And then they realized that this substance is called biotin. There are different names for biotin, vitamin H, or vitamin B7, or again, biotin. So we're gonna come back to this egg white injury again in a couple slides. So this is biotin. Of course, in all of these lectures, we tend to show a lot of molecules and chemical compounds, but don't get frustrated with this. Don't get confused because of course all of these things tend to make people a little bit anxious, but it's just to understand the function and how things work here. So the structure, the biotin structure consists of two different rings, here and here. One ring is a thiophene ring and the other one is a valeric acid. One ring is a thiophene ring, the other one is a urido ring, and both are attached to this valeric acid chain. Okay, we don't need to remember those words, even for me. They seem a little bit confusing. But it's important to keep in mind that usually biotin is binded to lysine. And when biotin is bounded to lysine, they form a compound that is called biocidin. So biotin and lysine, biocidin. Okay, So where do we get biotin? Of course, biotin is an important vitamin. We need to consume it from food. However, it's important to keep in mind that we can produce a little bit of biotin. Not exactly us, not exactly our bodies, but the bacteria that lives in our intestines, in our colon. So our microbiota, probably you've all heard a ton about microbiota because it's very popular nowadays. So our microbiota is able to produce actually biotin. But also, we also need to consume biotin through the diet. Again, liver is one of the main sources, one of the main dietary sources for biotin. I think I mentioned this a couple lectures ago, but liver is usually the main source for a really big amount of different vitamins and minerals, because of course liver is one of the most important organs in terms of storage for mammals in general. So 3 ounces of liver have 27 micrograms of biotin. Usually the recommended intake or the daily intake or the RDIs is 30 micrograms, and 3 ounces of liver has almost the entire amount of biotin that we need a day. Also eggs, specifically egg yolks. Egg yolks, one large egg or one large egg yolk has almost the entire recommended amount of biotin that we need to consume a day as well, 25 micrograms. Also bread, whole wheat bread, cheese, and Cheerios has none biotin, 0 biotin. And actually, you will see Cheerios in many other vitamins and minerals, because usually in this type of cereals, they come fortified with different vitamins and minerals. But apparently, biotin appear not to be fortified. Like, there's a lot of grain products and cereals that are not fortified with biotin. So in this case, Cheerios have 0 biotin. So when I mentioned a couple slides ago about the disease that came from eating raw yolks, that specific condition or disease is caused because of this compound here, avidin. I don't know how to pronounce that correctly. Avidin. So avidin is a substance that is present in raw eggs. It's a glycoprotein that binds irreversibly to biotin. It sticks to biotin and it won't detach from biotin by any means. So the bond between avidin and biotin is considered one of the tightest non-covalent bonds found in nature. The only way to detach these things is through heat. That's why we definitely need to eat our eggs cooked, because avidin prevents biotin from being absorbed and used by the body. So if we consume the eggs raw, the avidin that's present in the egg white is going to stick to the biotin that's present in the egg yolk, and it's going to prevent it from getting absorbed into our organism. Thus, it prevents us from receiving that biotin. Therefore, cooking eggs liberates biotin from avidin. I don't know if you ever seen, I don't know if it's still a popular practice, but a couple years ago it was fairly popular for like bodybuilders and people who drank those protein shakes, but instead of adding protein powder, they added raw eggs because they thought that raw eggs had more nutrients or more something in particular that I'm not exactly sure what it is. However, that's not a good idea in terms of biotin, because raw eggs can definitely substantially decrease the amount of biotin that your body is able to absorb. So when we consume biotin in foods, usually that biotin is attached to proteins. Usually we don't have biotin free in foods. It's usually attached to a protein. The digestion of biotin occurs in the small intestine, of course, and some proteases cleave biotin from proteins. So this reaction that happens in the small intestines yields free biotin and biotin. Biotin is biotin plus lysine. Again, we already mentioned that. So here we are in the lumen of our intestine. So if biotin is free, then we can absorb biotin through our enterocytes without any further digestion. However, if we have biotin binded to lysine, we need further digestion. So this complex between this union of biotin and lysine needs to get separated in order to be absorbed, of course. And who is in charge of that separation or that breakage on that bond is an enzyme that's called biotinidase. It's difficult to pronounce that. Biotinidase. So this enzyme hydrolyzes biotin in the small intestine. So this enzyme is in charge of separating biotin from lysine. However, this enzyme is very important for the absorption of biotin, but it depends on the pH of the intestinal juices to be activated. So we usually have a very stable pH in our intestinal juices. Usually it does not fluctuate much, but it can have some slight changes depending on different foods. And this pH is important because this particular enzyme acts differently depending on the pH of the lumen of the intestine. If it's too alkaline, it would do something. If it's too acid, it would do another thing. And here we have a pH scale, and different foods, of course, have different pH. Of course, lemon is one of the most acidic, and baking soap, of course, but baking soda is one of the most basic or alkaline ones. If our pH in our intestines is acidic, then biotinidase cleaves to biotin, and this produces what we want. This produces the result that we're hoping for, that is to free biotin from lysine. So both biotin and lysine are free and able to get absorbed into the enterocyte and then going through the blood and doing everything normal as it should be. However, if the pH is too alkaline, is too basic, is too high, then this enzyme, biotinidase, instead cleaves biotin, and then this enzyme becomes attached to biotin. Instead of freeing biotin, instead of making it free to be absorbed, it gets attached. So this attachment inhibits absorption. So this biotinidase plus biotin complex, this weird thing of both the enzyme and the vitamin together, is unable to get absorbed and then it's excreted. That's why the pH inside the intestine is so important to absorb biotin correctly. There's a specific situation where you can have a biotinidase deficiency. So you have a deficiency of the enzyme. You don't have the enzyme that is able to separate biotin from lysine. And this rare disease or situation was first discovered in 1983, and it's an autosomal recessive error of metabolism. So they saw that infants or children who were untreated for this disease developed severe biotin deficiency. Because of course, if you don't have the enzyme, you cannot separate biotin from lysine, then it cannot get absorbed, thus you have some severe biotin deficiency. Some of the clinical features of these biotinidase deficiencies include seizures, ataxia, dermatitis, and alopecia. So here you see a child with alopecia here with biotinidase deficiency and dermatitis here in his tiny cute feet. Similar to what we discussed a couple lectures ago when we talked about riboflavin, the absorption in the enterocyte of these particular vitamins is dependent on the concentration of the vitamin. So if we have a lot of vitamin, a lot of biotin in the lumen of the intestine, then it's going to get absorbed through passive diffusion. So it's going to get absorbed without needing extra energy. But if we have a low intake or there's a low concentration of biotin in the lumen of the intestine, then we will need a specific transport to absorb biotin inside the enterocyte. In this case, it's a sodium-independent transport. Again, I'm still going to reference a lot about vitamin B2. Alcohol has an important role in the absorption and metabolism of biotin as well. So alcohol inhibits biotin absorption and metabolism as well. We're not exactly sure what's the exact mechanism but why alcohol inhibits absorption of biotin and also riboflavin. And there's been some studies in humans that have seen lower circulating biotin in alcoholics or people with severe alcoholism. And there's also been some animal studies that have seen that when animals are administered high doses of alcohol, absorption of certain vitamins, including biotin, is altered. Now, remember that I mentioned that biotin is not only able to be absorbed, is not only able to be obtained from food sources, but it also can be synthesized by our own microbiota that is present in our colon. Well, these microbiota can produce free biotin, and this free biotin then gets absorbed using a sodium-dependent transporter or carrier-mediated and sodium-dependent transporter into the colon and then into circulation. So, in simple words, the biotin that's produced by our microbiota can get absorbed in the colon and then it goes to our blood or circulation and is able to reach all our tissues. How is biotin transported in our blood? Actually, biotin is able to be transported free. There are very few compounds in our body that are able to be transported as they are, like free in the blood. And biotin, apparently, I honestly had no idea about this, I just learned it through this lecture, is able to travel freely through our blood. So 80% of biotin travels in free form in our bodies. And there's a minor percentage of biotin that is bounded to albumin and that is bounded to alpha and beta globulins. So we already talked about how biotin is digested, is absorbed in our intestines, dependent on pH, how it is transported in the blood. And now we need to know or to learn how biotin uptake into tissues, how it is how we are able to uptake biotin in the different tissues of the body. So there are small amounts of biotin that get stored, and the key storage sites of this biotin is in the muscle, liver, and brain. And the biotin that is present in our blood, thanks to SMVT, can then pass through our muscle, our liver, and brain. We can either use biotin in these different tissues, or we can store a little bit of this biotin as well. So now we have to learn the functions. Of course, we know what biotin is, we know some deficiencies, we know what happened, we know that some alopecia, ataxia, etc. But we actually don't really know so far what are the exact functions of biotin? Why do we need to consume this vitamin, or why is it so important for us? Again, these vitamins have some very important coenzymatic roles. As I mentioned on that lecture a couple lectures ago, most vitamins have very important roles as coenzymes. In this case, one of the most important enzymes that biotin acts as a coenzyme is pyruvate carboxylase and acetyl-CoA carboxylase. But it also has some other functions independent of the coenzymatic role, such as cell proliferation and gene expression. So now we're going to talk about the coenzymatic roles. The coenzyme form of biotin is called biotin coenzyme, of course, makes sense. Biotin is bound to the lysine residue of carboxylase, and biotin functions as a carrier of activated carbon dioxide. And this is the most important thing to learn and to remember about the coenzymatic action of biotin. The main role of the coenzymatic action of biotin is putting or carrying an activated carbon dioxide molecule into a specific enzyme. So here we have the activated carbon dioxide. So whenever you see a carboxylase or something that resembles a name carbon dioxide, you need to think about biotin, because biotin is important on those reactions in specific. So one enzyme where biotin is very, very important is pyruvate carboxylase. Again, carboxylase has something to do with carbon dioxide, and biotin is important on that reaction. This one catalyzes the carboxylation of pyruvates to form oxaloacetate. Okay, this sounds complicated. Probably you're all tired in this point of the lecture, but it's important to remember and to put this in context. So pyruvate is the end product. So let's say in a more simplistic way. When you use glucose to produce energy, one of the midpoints of that reaction from glucose to energy is the formation of pyruvate. And in order for pyruvate to go to the Krebs cycle, and the Krebs cycle is almost the last step of the production of energy from glucose, pyruvate needs to be transformed into oxaloacetate. And actually, our beautiful coenzyme or vitamin is in charge of transferring activated CO2 to a reactive carbon of the structure, thus inducing the transformation of pyruvate to oxaloacetate. So it activates this specific enzyme that transforms pyruvate to oxaloacetate. And then oxaloacetate enters the Krebs cycle. Probably you already learned about the Krebs cycle. Maybe you don't remember. I don't think that there's people that actually knows the entire Krebs cycle. I keep on knowing the Krebs cycle, and then I forget about it, and then I learn it again, I memorize it again, and then I forget it about it again. So it's like one of those things that if you're in this field, you're going to encounter it several times in your career, but you're never going to completely memorize or learn it. So oxaloacetate enters the Krebs cycle on condensation with acetyl-CoA. So this enzyme, the enzyme that biotin activates, has a very important role in our metabolism because it regulates what happens. So if we have a lot of energy in our cells, so if we have a lot of ATP in our cells, then this oxaloacetate, instead of going through the Krebs cycle, it goes to an opposite route that is called gluconeogenesis. And gluconeogenesis, forming glucose from something else, in this case oxaloacetate. So we will form glucose from this oxaloacetate. Because we already have energy, we don't need more energy, we don't need more ATP, we need to store things. So we're going to send this oxaloacetate to gluconeogenesis, and maybe we can do glycogenesis after that, so we can store glycogen in our liver or muscle. However, if we have just a little bit of ATP, if the amount of ATP in our cell is low, meaning we don't have enough energy in our cell, meaning we need energy, then this oxaloacetate do enter the Krebs cycle. Instead of going to gluconeogenesis like we mentioned before, no, it goes to the Krebs cycle, because the Krebs cycle is basically one of the last steps to making a ton of ATP for our cells, and ATP is energy. So the enzyme that is in charge to making this decision is the pyruvate carboxylase, and pyruvate carboxylase is activated through biotin. So here we can see the importance of the coenzymatic form of biotin in the regulation of metabolism. So we talked about pyruvate carboxylase. So this is everything related to Krebs cycle, pyruvate, glucose. This one is a different story. This is another enzyme. And this enzyme is called acetyl-CoA carboxylase. And this enzyme, instead of having something to do with oxaloacetate or with pyruvate or with the Krebs cycle, blah, 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 this one has to do with fatty acid synthesis. So this enzyme catalyzes the formation of malonyl-CoA from acetyl-CoA. So this sounds a little bit confusing, but basically, in simpler terms, this enzyme initiates fatty acid synthesis. So whenever we have too much acetyl-CoA, whenever we did, maybe it could come from glucose, like for instance, someone ate too much carbs on a certain meal and it has too much glucose, and we have no idea what to do with that glucose. We cannot store it in the liver or in the muscle because we already have our storage full. Then our body says, okay, instead of storing that glucose as glycogen, we can store it as fat. So we can transform that into fat. And biotin is required by the enzyme that initiates this process of transforming acetyl-CoA into like basically fatty acids. So biotin transfers an activated CO2. Again, remember, this is one of the main functions of the coenzymatic form of biotin. Transfers an activated CO2 to a reactive carbon on the substrate. And this reaction commits acetate units to fatty acid synthesis. So all of these complicated wording, complicated images is basically saying that biotin, the coenzymatic form of biotin, is very important whenever we need to form or to create fat molecules or fatty acids, whenever we need to transform something into fat so we can store it in our adipose tissue. So those are the coenzymatic roles, or the roles of the coenzyme forms of biotin in acetyl-CoA carboxylase and pyruvate carboxylase. So those are the coenzymatic roles. There are some others coenzymatic roles of biotin, but these are the most important ones. Now, we also mentioned that biotin have some non-coenzymatic roles. Some roles that have nothing to do with the coenzyme or with activating enzymes or not. One of those is that apparently biotin have an effect on cell proliferation. We're not exactly sure what's the exact mechanism about why biotin can have an effect on cell proliferation, but apparently it could be because biotin can attach to histones. Histones are some tiny proteins that are bound to DNA as part of chromatin, and when biotin binds to these histones, this binding apparently can increase cell proliferation. And cell proliferation can have an effect on all of those tissues that are constantly renewing, such as hair, such as nails, such as skin. And that's why probably biotin is marketed as a supplement for having beautiful hair, beautiful nails, beautiful skin, because it has an effect on cell proliferation, among other effects, of course. But again, we're not completely clear about the exact mechanism or about why this happens. Also, biotin has a role on gene expression. There's more than 2,000 genes, human genes, that depend on biotin for expression. Biotin appears to be necessary for translation of these genes. So biotin basically can activate the transcription of certain genes. This is the metabolic route. We're not going to go into too much detail here, but it's important to keep in mind that there's a big amount of genes that are dependent on biotin to get expressed. If biotin is not there, then those genes won't get expressed as they are supposed to. So we already talked about how biotin is digested, is absorbed, is transported in the blood, how it is absorbed into different tissues, the different functions that biotin has in different tissues. And now we need to know, how can we excrete biotin? How do we get rid of biotin? So first of all, biotin usually, when it's functioning, it's binded to carboxylase and to lysine. So just like we need biotin by itself, alone, free to be absorbed, we also need biotin by itself, alone, to be excreted. Sorry. So we need to remove the carboxylase group and the lysine group from biotin in order to get to excrete it correctly. So an enzyme that's called a protease is going to remove the carboxylase part, and then again, our good friend, the biotinidase, the same enzyme that we talked about when we discussed about pH in the lumen of the intestine, blah, 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 blah. Again, this enzyme removes the same thing, the lysine from the biotin, freeing biotin and making it able to get excreted as it's supposed to. How do we excrete it? Most of the biotin is excreted through the urine. So 98% of biotin is excreted through the urine, and it can be excreted as biotin or as its metabolites. And metabolites is when biotin was broken down for a specific reason. And we also can excrete a little bit of biotin through our feces, because remember that our bacteria can form or can synthesize biotin, and if for some reason that biotin that was formed by our bacteria was not absorbed correctly, then it's going to be excreted on our feces. So the adequate intake of biotin, we already mentioned this briefly, but in infants is around 6 micrograms a day, children 12, males and females 30, pregnancy 30, lactation a little bit higher, 35. So the number that you might need to remember the most is this 30. So 30 micrograms a day is the adequate intake of biotin. What happens if we don't have enough biotin? There are different reasons that we might get deficient. We're going to discuss this in a while. But if we are deficient in biotin, we might experience lethargy. So we're tired. We don't want to perform our usual activities. We might get depressed. People usually experience muscle pain. Hair loss is a very common one. So deficiency of biotin is usually linked to hair loss, and it could be because of this cell proliferation function that we mentioned before, and scarlet red dermatitis. The main biotin deficiency disorder is the egg white injury that we mentioned when people drink or take or eat raw eggs. And it's characterized by hair loss and dermatitis. And which population is affected by or could be at risk of deficiency? First of all, the people that consume large amounts of raw egg whites. If you consume 1 or 2 raw eggs or raw egg whites, it's not going to be that detrimental. But if you consume more than, let's say, 5 raw eggs a day, then you're at higher risk of biotin deficiency. People with inflammatory bowel disorders, with some sort of absorption difficulty, short bowel syndrome, uncontrolled celiac disease, people with bariatric surgery that removed a big portion of their intestines, etc. Anyone that can have problems with absorption can be at risk of biotin deficiency. We also mentioned about people with severe alcoholism that can alter absorption and metabolism of biotin. Also, smokers catabolize or use biotin faster, so their needs apparently are higher. People that experience prolonged fasting, of course, because they do not consume the amount of biotin that they need. People with anorexia nervosa, etc. Biotin status may decline during pregnancy, and this is something important to keep in mind. 33% of pregnant women might be affected by this biotin decline during pregnancy. It occurs due to increased urinary excretion of biotin during pregnancy. For some reason that we're not exactly sure what's the mechanism, some pregnant women tend to lose excessive amounts of biotin through their urine. And these, in some studies, that they're not like RCTs, this is not experimental studies, these are just associative findings, but they've seen that there's a relationship between deficiency of biotin during pregnancy with higher incidence of babies that are born with cleft palates. So there is a relationship between biotin deficiency and cleft palates on the newborn, on the baby. Also, biotin supplementation during pregnancy may prevent this. So it's important to keep in mind that some pregnant females should supplement with biotin. The normal intake, again, is at 30. That number is, I think it's the most important number to keep in mind. And again, no tolerable upper limit has been established because it's a water-soluble vitamin, and the excess, we remove it from our bodies through the urine. Intake of up to 100 milligrams for several months has not reported toxicity. And keep in mind, 100 milligrams, where the normal intake is 30 micrograms. So this is more than 3,000 times the adequate intake. And these really high doses are given to people with inherited disorders of biotin metabolism, like the ones that we mentioned a couple slides ago, those people that do not have the enzyme that separates biotin from lysine, or other disorders. And again, doses really, really high have not reported toxicity. Also, very high doses of biotin are used therapeutically, such as diabetes. So people with diabetes, there's been some studies that have shown some benefits of biotin in glucose control. So there's been some cross-sectional findings in people with type 2 diabetes that have lower plasma biotin levels and higher plasma glucose. So because these are cross-sectional studies, we're not sure what's the relationship of the causality. Is it that a low plasma biotin induces high plasma glucose, or is it that high plasma glucose induces, for some reason, low plasma biotin? We're not sure because, again, this is cross-sectional studies, observational studies, and we cannot assess causality when it's just cross-sectional, like an observational study. However, we have some clinical trials, and remember with clinical trials, when you have a control group and you have an intervention, it's an RCT, you can assess causality. So biotin supplementation may help to decrease fasting plasma glucose level, and this has only been observed in participants or people with diabetes. For instance, this clinical trial, people with type 2 diabetes versus healthy controls received 9,000 micrograms of biotin a day for 4 weeks, and they saw that fasting plasma glucose decreased by 45% in those patients that had diabetes. Another study with participants with diabetes and healthy controls, they supplemented 15,000 micrograms a day of biotin for 4 weeks, and they saw no effect in either group. And there was another study with type 1 diabetes. This is different. Again, this is type 2, and this third study is type 1 diabetes versus healthy controls again, and they gave 16,000 micrograms a day for a week, and they saw that fasting plasma glucose decreased. We have no idea the exact percentage, but decreased in those with diabetes. Not in the healthy controls, but in those with diabetes. So apparently, there is an effect of biotin in glucose regulation in people with either type 2 diabetes or type 1 diabetes. What is the mechanism of action? We're not completely sure, but it could be one of these three. The first one, or it could be all of them. So the first one is that biotin increases the utilization of glucose for fatty acid synthesis. A couple slides ago, I showed you, or I told you, that biotin is important in activating one enzyme that transforms basically glucose into fat. I'm saying this in very simplistic words. So if biotin is important in transforming glucose to fat, that means that the cell is able to uptake or to clear glucose from the blood more efficiently. And if you're able to clear glucose from the blood because you're going to transform it into fat more efficiently, then that means that you will end up with lower fasting glucose levels. Also, biotin stimulates glucokinase activity, and glucose kinase activity increases the synthesis of glycogen, and this is the storage form of glucose. And again, if you are increasing the storage of glucose, then you're also being able to catch more glucose from the blood, then you're clearing glucose from the blood more efficiently, and if you clear or if you uptake more glucose from the blood more efficiently, then you will result with lower fasting glucose levels. And the third option is that biotin stimulates the release of insulin, but we have only seen this in animal studies. But apparently, biotin stimulates insulin, and if you stimulate insulin, insulin is able to basically catch glucose and move it into the cell, again clearing glucose from the blood more efficiently, so resulting again in lower fasting glucose level. So even though we don't have enough data, we have some information and some possible mechanisms by why insulin could be a sort of a therapeutic alternative for people with diabetes to make glucose disappear from the blood more efficiently or get accepted into tissues more efficiently, lowering fasting glucose levels. There's another situation where biotin can be used as a therapeutic agent, and it's called brittle fingernails. This photo makes me very anxious. I don't know why. I hate everything related to nails. So there's been a couple clinical trials, but they've been uncontrolled, so without a control group. So we have 3 uncontrolled clinical trials that have been published, usually in female subjects between 25 and 55 years old, with small sample sizes, like just 10 people per study, so it's like tiny studies. But they've given participants biotin supplementations 2.5 milligrams a day. So this is again a high intake, because remember that the daily allowance or intake is just 30 micrograms. And this is 2.5 milligrams a day. for 6 months. And they saw that 75% of subjects reported clinical improvements in their breeder fingernails with the supplementation of biotin. The fingernail thickness increased by 25% measured by electron microscopy. However, of course, we need larger placebo-controlled studies to make solid conclusions. But this is interesting, and it could be due to the cell proliferation function of biotin. So this is a mini review. You've already discussed about vitamin C, thiamine, riboflavin, niacin, vitamin B5, B6, and now biotin. We're not going to go into too much detail with the previous ones, but we are going to mention that biotin is present in meats, some grains, and we also can produce biotin through our microbiota, our own bacteria that lives in our colon. The main function is to transfer CO2, but it also has some functions in cell proliferation and gene transcription. The deficiency is called egg white injury when people consume raw eggs. And again, there's no toxicity because it's a water-soluble vitamin. Some water-soluble vitamins can have toxicity, but in this case, there has not been toxicity established. So thank you so much for listening to this class. I'm so sorry if something was confusing. I don't know if I pronounced all the words correctly, but if there's something that was not clear or that you have doubts, again, you can ask. Probably there's a certain part in the Blackboard thing where you can leave the questions. I'm not sure how that's set up, but if not, you can email me to my email that is scienf, so SCIENF2@uic.edu. So thank you.